Hello everyone, I am Mohammad Arifur Rahman. I am a PhD candidate in computer science at George Mason University. Today I'll be presenting our paper IDMIL, an alignment-free interpretable deep multiple instance learning for predicting disease from whole metagenomic data. My co-author is Dr. Josefa Rangwala, professor in computer science at George Mason University. This is an overview of my presentation today. I'll be talking about metagenomics and disease prediction, multiple instance learning, some of the related work, our proposed IDMIL with all the steps, the dataset we have used, the results and comparative analysis, sensitivity analysis, followed by conclusion. So the specific aim for this project is to predict disease from whole metagenomic data. What do we mean by that is, given a human subject, we have access to the metadata. For example, if the person is suffering from a certain disease, like liver cirrhosis, IBD, diabetes, or if the person is healthy at all. We also have access to the metagenomic sequence data, which is subjected to heavy preprocessing. All this data works as a input data to a machine learning unit. So we train a classifier, and if we give new data, our expectation is that the trained model should be able to predict what is the class level of the person, whether the person is healthy or unhealthy. We also want to predict which microbes are likely to be correlated with the disease. So that is the interpretation part. In this workflow, these are the three steps where statistical and machine learning involves. And we'll try to solve this problem using multiple instance learning combined with deep convolutional neural network. So in a typical supervised learning mechanism, we have labeled data, say these are positive data and these are negative data. We train a model, and if we pass an unlabeled data to this model, the model should predict which one is positive and which one is negative. So in multiple instance learning, the data is not just one single vector or data instance. Instead, the data is composed of multiple data instances, which are collectively representing that sample. So in MIL, we call this sample as bag, and we call the constituent parts as instance. Usually we have labeled bags and unlabeled instances, which is represented here as question mark in the diagram. So let's say we have positive bags and we have negative bags. We train an MIL model. Given unlabeled bags and unlabeled instances, the MIL should be able to predict which bag is positive and which bag is negative. On top of that, MIL should also be able to predict which data instances are correlated to the bag level. In other words, which data instances are likely to be positive and which data instances are negative. So the later one is preferable in our scenario. Here I'll be presenting some of the related work in this area briefly. So there are research work that studied the relation between different types of disease and the shift in microbial diversity. For example, the obesity, oral disease, neurological disease, Crohn's disease, and so goes on. One of the popular methods in this area is MetAML, which involves identifying the marker genes and species level abundance as features, and then using a random forest as classifier. So this method uses reference sequence database of known microbes, which can limit our ability to identify novel microbes if there exist any. It discards lots of sequences, so the sequence tree that does not get any match from the reference database, those are discarded, and that involves information loss. The errors in the profiling process also propagates to the later analysis process. MIL was first proposed by Dieterich back in 1997 for drug activity prediction from molecular steps, which provided better accuracy as well as instance-level interpretation. One of the earlier work in this area was Misvm, which simply modifies the standard support vector machine's maximum margin formulation. It also used the standard assumption, which is negative bags will only contain negative instances and positive bag will have at least one positive instance. This assumption is also very restrictive for practical purpose. So SBMIL, it relaxes the assumption of MISVM by saying that in positive bags there will be few positive instances. On the other hand, GICF, it relaxes that assumption even further by saying that negative bags will contain some positive instances. making GICF more practical for other problem setups because it allows for bags to have both positive and negative instances. Attention-based multiple instance learning is one of the recent work that combines MIL and deep convolutional neural network. It applies neural attention to create bag-level representation. And the issue with that is the mapping from hidden layer to the input layer is lost. You cannot interpret the result directly. Also, it was primarily proposed for image data. PLG-ABD, it identifies the species level abundance and sorts them based on biological taxonomy. It creates a heat map from the taxonomy and uses those images as a representation of the metagenomic sample, followed by a deep convolutional neural network for classification. It has similar drawback as MetaML for using the reference sequence database and not utilizing the metagenomic data. Also, it lacks interpretation. So the first step of IDMIL is to create the KMR embeddings. So given the context of a KMRI, which is simply the combination of prefix and the suffix of KMRI, we try to maximize the probability that KMRI appears. In the process, we learn this vector representation of KMRI. This is something similar to a very popular method in natural language processing called the word-to-vec. We also use pruning technique to remove some of the unnecessary KMRs from each sample. So we use this TF-IDF technique where WXI represents the weight of KMR X in any sequence read Y. So TF term is simply the frequency of X in any sequence read, and the right-hand side term provides the relative importance of any KMR X in that particular sample. So if the frequency of KMR is high, then this score will be higher. On the other hand, if a KMR appears in almost all the sequence reads, maybe due to the sequencing errors, then it will receive a lower score. So combining these two, we get a scoring for each of those KMRs with respect to a sample. So we take the top 70% of the KMRs. So now that we have the KMR vectors, we use those to create the sequence vectors. So in order to do that, we simply take the KMR vectors in each sequence and we use this power mean formula to create the sequence vector. The interesting fact about power mean is that by changing the values of P in this equation, you can get interesting statistics. For example, the minimum, maximum, geometric mean, arithmetic mean, and so goes on. So we chose the minimum, maximum, positive and odd values, and smaller values. So altogether, there are 4 values of P used in this study. The good thing about this is there is no extra parameter or learning weight in this method. So RNN, LSTM, and all the other sequence embedding approaches in using neural network, they have scalability issues when applied on raw whole metagenomic data with millions of sequences. Here in this approach, there is no learning involved. It is quite fast and parallel in nature. At this point, we have the sequence vectors, and we use those to create our instance vectors. To create the instance vectors, we use miniVeg k-means in each sample in parallel. So miniVeg k-means is a variation of k-means, but it optimizes the same objective function, and it uses miniVeg to gain computational efficiency. So we increase the number of clusters as a power of 2 as long as the cross-validation performance keeps improving. So the higher the number of clusters, the more variational information you get. However, it also increases the computational expense. So now we have the instance vectors. We create the bag representation for each metagenomic sample. To create the bag representation, we simply create a reference point, href, which is the average of all the instances in the training set. And then we sort all those instances based on their corresponding distance from the href in ascending order. So that means if we create a matrix based on this ordering, then similar instances will be closer to each other and dissimilar instances will be further from each other. This directly practices the triangular inequality of the distance metric, and it creates a locality within the instances, which in turn creates the premise for our convolutional operation, which will be used later. So this is the classification part of IDMIL. On the top left corner, we have the bag representation. The first thing we do is apply positional attention values. which is simply a softmax equation here. So that means the positional attention values will be in the range of 0 to 1, and they will sum up to 1. We multiply these attention values with each row element and then restructure the data. We apply one-dimensional convolution so that it conforms with the ordering we introduced earlier. After multiple hidden convolutional layer, we bring it to a fully connected layer and predict the class table of the bag as either healthy or unhealthy. So once we are satisfied with the cross-validation performance, we investigate the positional attention values. So we see that which positions are taking the highest attentions. We then investigate which clusters are taking those positions in the bag matrix from the patient samples. And we then go back further and investigate which sequence reads are actually the cluster members. So the ones with higher attention values will be indicative of the disease, and we should investigate those sequences, and the ones which are not very important for subsequent analysis. So these are the data sets that we have used in our study. We have used the liver cirrhosis dataset, colorectal cancer dataset, inflammatory bowel disease, obesity, and type 2 diabetes datasets. So the minimum number of average sequence per sample here is 40.2 million. So that shows how big these datasets can be and how important scalability and efficiency is in our project. So these are the results of IDMIL. We can see that IDMIL outperforms other approaches for all the datasets in terms of average AUC-ROC values. So the reason can be inherent in the steps of IDMIL. For example, the TF-IDF score that we used in the earlier step, it removes the uninformative k-mers and noises from the system. The instance clustering removes the effect of repetition in the sequence read of the whole metagenomic data. The neural attention, it suppresses any noise or noisy input by multiplying it with near zero values before the CNN starts. And the CNN is effective because by setting the number of output channel in the CNN model, we can actually force the model to learn more complex variation that can be missed by other statistical and machine learning approaches. So all this together helps IDMIL to perform better in terms of predictive performance. So these are the sensitivity analysis performed in this project. We wanted to see how AUC-ROC value changes with the choice of k-mer length, the embedding dimension, and the number of cluster. So you can set a cutoff value after which the computational burden increases significantly, but it does not provide any significant increase in AUC-ROC value. So for example, the k-mer length was set to 10. We have chosen the embedding dimension as 2 to the power 6 or 64, and we have chosen the number of cluster per sample or instances per sample as 2 to the power 14 or 16,384. So this is a way to interpret the result from IDMIL. We went back to the original raw sequences and we found out all those clusters of raw sequences that are getting higher attention values. So on the left hand side, we have this graph of identified microbes from blood sources with their corresponding attention values. So we have that graph for liver cirrhosis data, and on the right hand side, we have the graph for the colorectal cancer dataset. And we have found out that some of those renowned pathogens and microbes, which are found to be correlated to the disease by the current microbial studies, have also been identified by IDMIL with higher attention values. In conclusion, this paper shows an application of multiple instance learning in the domain of metagenomic analysis. We have shown how to implement the multiple instance learning formula using deep convolutional neural network. We have also presented an efficient and effective data representation from raw metagenomic sequencing to the multiple instance learning. So we have provided a way to interpret the result that can directly go back to the raw sequences and perform all the profiling on limited number of sequences. This method has potential for future improvement as well as other applications. Thank you very much. Your questions and suggestions will be appreciated.